All right. Hi, everyone. Some of you, good morning. Some of you, good afternoon, right? I'm in good morning. I have 11 a.m. right now. So I am going to present some of my work here based on the platforms we have developed in last 5 years in the lab. I started at SIU in 2015, um, submitted my package, and just finger crossed, let's see what's going to happen. So working on Vexon plastic platforms for point of care diagnostic applications. So just before heading on to the main presentation, just a quick advertisement. So we are in the Carbondale, which is just 2 hours from St. Louis, Missouri, and we had a full solar eclipse in 2017. And we are gonna have another one full solar eclipse on 2024. Just heads up, maybe in case you just wanna stop by and then watch it. So just put in your calendar, right? So that's I just want to mention very quickly. These are the past members of the group in the last 5 years. Fantastic people, wonderful minds and hands. They helped me a lot to develop some platforms. And currently I have like 4 graduate students. One of them is getting PhD this spring and graduating. And I have like a lot of good collaborators. I'm really proud of working with them. Especially I want to thank Professor Lisa Ellerby from Buck Institute and Keith Gagnon from SIU School of Medicine to give us samples, because we are more kind of like bioanalytical or analytical chemists working on proof of concepts, and we get real samples from different sources and our collaborators. And funding, definitely dollars are important. Got some NSF funding and recently from NDR, which we are really excited about. So let's have a step back and think about some of the tools we are using nowadays in our houses or maybe in the clinics. So like glucometer, you know, everybody knows about it, alcohol analyzer or drug abuse analyzer, and Abbott iStat. So these are the equipments nowadays in the market, and they are pushing the boundaries and helping people in point of care diagnostics. So you can buy glucometer for less than $50 maybe, or around that cost, and Abbott iStat, which is about $6,500, right? So it is not like in the hands of like end user, but you can find this in clinics for point of care diagnostics. And a pregnancy strip, which is one of the simplest kind of like litmus test kind of thing, or litmus test kind of biosensor for yes or no answer, right? And why these point of care diagnostic tools are really important, and they are getting popularity because they are simple, they are rapid, you can get them portable devices. Some of them are really sensitive, and they still need to like improve their sensitivity, and they're selective, and especially this is because of the low cost, like for the glucometer or alcohol analyzer for sure, like can be used by people, you know, a layman, right? But it is not for the Abbott iStat, which is used in clinics because of its cost. So so point of care testing, it's getting popular, and lots of companies are in this market. And one of the reason is they're not like fast, portable, or accurate or affordable, but also it helps physicians to for the rapid triage and treatment decisions. So that's how they can make a timely decision for the diagnosis as well as the treatment, and also it can help them monitoring treatment response. So patients, they can look at the patient condition real time, and they can monitor the treatment response using these point of care diagnostic tools. So these are the like reasons because of point of care testing is getting popularity, and And lots of companies are in market right now. You can just see on the right-hand side, from Roche to Nova Biomedical or Quiddle or Abbott Laboratories or Siemens. All of them are in this market. They are developing their own tools. They are developing new tests, new methods, protocols, devices, capabilities, so on and so forth. And the market by this year is expected to be $30.45 billion, right? And with 3.25% growth, that will be perhaps $35.74 billion by 2028. And in market terms, it is called as product outlook, but in our language, that will be like type of testing or samples or diseases, detection. So there is a long list from glucose testing to fertility or cardiac biomarkers, hematology, or lipid testing, cancer biomarkers, and drug abuse or urine analysis, right? So there are a lot of like different type of samples you can analyze. There are lots of different kind of tests available. Based on that, like doctors can make their decisions, or they can see a real-time response of the patient. But in all of this, what is missing is point of care tool for neurodegenerative diseases. So there's a niche, there's a need, there's a space for the tools to be developed to detect neurodegenerative diseases. So the tools, which are quite familiar nowadays, like glucometer or alcohol analyzer or Abbott iStat for detecting like different biomarkers in blood, they're actually a combination of these three fields, like microfluidics, electrochemistry, and biosensors. So these tools were developed based on the interfacing of these three different fields. And I will just have a very quick overview of these three fields, and we'll move on to my own results to help neurodegenerative community. So microfluidics is actually based on the concept of lab on a chip. What it means, like you develop a chip size of credit card, or maybe smaller than that, like 2 centimeter by 2 centimeter, or maybe a little bit larger or smaller. And the main purpose is you want to perform major laboratory procedures on a single chip, and you want to handle volume down to microliters and nanoliters. So that's how you're going to reduce the footprint of a lab. You're going to reduce the size of the sample. You're going to save money on the reagents, and you can have a mobile laboratory rather than a centralized facility, right? So that is the whole concept of the lab on a chip and microfluidics. And based on that, you can develop miniaturized like platforms like these cartridges, which you can use for handling sample, like this strip of the glucometer, where this small strip where you can drop your blood, and the sample size may be in microliters, and then that chip can handle your sample and give you a simple response on the readout. So you also want to develop some miniaturized platforms for sample preparation or sample handling, as well as the detection, and with the capability of interfacing with portable readout devices. So you have a small platform which has a low cost, which has a low footprint, which not only handle the sample, it can also detect and as well as interface with a readout device. So there are 4 different kinds of microfluidic system. One which is like the microfluidic field started with was the channel-based, like you have sophisticated channels in PDMS or polycarbonate material, and these channels are run by several different pumps. So the chip size is smaller, but overall footprint is large because you need a lot of pumps to run these channels. This digital microfluidic, where I worked like for several years before SIU, and this is based on applying potential. So this looks like cool because it just looks like a video game, like you applying potential from electrode to electrode. You can program it, and then droplets move the space where you want. They can mix, merge, they can split, they can dispense, and you can detect at the end of the chip or maybe at the center of the chip, depending on your sensing tool, right? One of them, the low-cost microfluidic system is paper-based, which just got like popularity in the last 10 years when it emerged from the Whiteside's group at Harvard. But paper-based, microfluidics, in other words, the chromatography was first developed on paper, right? So it is not just recent, it was there for the last 50 years, but got popularity because of the microfluidic field, which just emerged in the last 20 years. And now we develop like plastic-based microfluidic systems because plastic is more robust and tough than paper, and then you don't need external forces or external driving actuators which drive your sample, right? So the sophisticated systems like this channel-based system or digital microfluidic system, you really want to have a facility which is known as clean room facility, right? Where you want to, you have to have specific suiting, training, equipment, clean environment, which give you high resolution devices which are also sensitive, right? But the downside is they are expensive and the work is laborious, right? So even a small mistake can just ruin the whole batch, and that batch of the devices might cost you like several $1,000s. So recently, the flexible microfluidic system of flexible devices, they have emerged, and the market share is going to be $10.5 billion by 2022, which is one third of the market share of the whole point of care diagnostic devices, and which you can print on different substrate, on paper, plastic, or different kind of materials, just using printing methods. So maybe they are not as sophisticated as the clean room based devices, but they are better in case of like you have a low cost. If you ruin the batch, it's not going to destroy the whole lot of things, right? So so that was about the microfluidic systems and then flexible devices and their market share. Now I will move on to electrochemistry, which is one of the oldest analytical technique, which we have been using for one way or the other. And over the years, the size of the equipment, right? So this duck-shaped cyclic voltammetry is famous. It is a symbol of electrochemistry, but although the electrochemistry is not about the cyclic voltammetry, but over the years, the hardware of the equipment has significantly reduced just based on the Moore's law, right? So Moore's law is saying exponentially number of transistors are increasing, which will cause decrease in the size of the equipment. So that's what we see, almost 1/3 of the size of this old EG-NG potentiostat. Now we have CHI, like 1/3 of the size, the cost also reduced. Now we have a potentiostat, which is almost 1/10 of this CHI system. So this is maybe PalmSense. And now we still have, like, we have further smaller systems, which is down to the size which you can hold in your palm, right? The functionality might be a little compromised in terms of the range of the current you can handle, but still it is low cost, it is handy, it is portable, and you can also insert a chip into this potentiostat. So not only the hardware itself, but the size of the sample has also reduced over the years. So this moved down like from several hundred milliliters to this 20 milliliter vial, and from 20 milliliter vials to this small chip, right? So you can handle the sample size down to maybe 5 microliter on this small chip, and we can make this chip in our lab just by printing methods. So this is from like the lab we have developed in our lab. Now you can just look at the whole thing, your potentiostat, your sample connector handle, your chips, your tablet, your computer, and everything can come just in a small briefcase. And the cost will be like almost 1/10 of the cost of the potential stat that used to be like 30 years ago, right? Now I will just introduce about the biosensing field, just in case if you're not really aware of that. So biosensing is just based on a very simple concept that you have a transducer surface, a solid surface of a transducer, which can be an electrode or some other surface where you can immobilize your bioreceptor layer like antibody, and you bring your sample, your antibody is selective to this target, it captures the target, and then you see a signal as a result. This signal can be change in color. This signal can be change in the property of the transducer. This signal can be movement of the electrons. This signal can be change in the frequency of the transducer. So there could be like a whole lot of different change on the transducer surface, which you can measure as a result of this binding event, right? So you can capture like cells or proteins or nucleic acids or small molecules like drugs or prions. So you can develop a whole lot of different kind of biosensing strategies for different analytes or biomarkers. There could be a label system where you have a label like an enzyme which catalyzes the process or catalyze the binding event, which result in a sensitive signal. That signal can be again change in color or fluorescence or the current production. And there could be another strategy which we call as label free. So there is no label attached to it. What all it about is change in the property of the transducer. So as a result of this binding event, the property of the transducer changes. For example, its resistance will increase, as you see in this plot EIS. So resistance of before binding is lower and the resistance increases as a result of binding, or the frequency might change as a result of this binding. So this is a label free. So, so what it means, you can develop different kind of biosensing platforms, methods, protocols. You really need to work on sensitivity, selectivity, and you know, the complex metrics. So this slide is just about sort of like, I will say, marketing from our lab. So we accomplished like tons of, we have applied different kind of electrochemical techniques for biosensing applications, from voltammetry to electrochemical imaging. You can image a surface based on electrochemistry. And here you see a difference between, this is a mismatch sequence, mismatch, and this is a match DNA sequence. So there is a difference in the topographic image of these sequences, which you can image by electrochemical technique. And then we also can incorporate into microfluidic system. So we can perform these kind of detections in a simple microfluidic platforms by incorporating in electrochemistry and small chips. So now we have moved on to printed devices, because printable devices, they are cheaper, easy to fabricate, simple. The downside is they are not as high resolution like the one you can prepare in a clean room. But again, the one we have prepared using these 3 printers, like this is a wax printer. Maybe you don't know this, you're using is a wax as an ink here. This is inkjet printer. And this is a polyethylene terephthalate substrate. This chip is based on only these 3 components. And the size of the electrode is down to like 1 millimeter. Right, so you can reduce the size just based on these simple printing tools. And we also make some inks, a sensitive material ink. For example, this is molybdenum sulfide ink we developed, which the cost is about $10 per 5 mil. If I want to buy this from Sigma, it's going to cost me a couple of $100s. So, so the crux is this printable system has a market. It has feasibility. You can build your own system. You can develop your own protocol and design your device in a real time. And you can modify based on your need and apply on your target. So so we have developed over the years several different Vexon plastic platforms. Vexon plastic, why I call it Vexon plastic, because we make a template based on this Vex printer and on this plastic template. Why plastic? Because it is tougher than paper. The sample is not absorbed by the substrate, which is happening in the paper. So that's why it's beneficial. You can do different kind of detection. You can incorporate different kind of detection tools on this substrate. So this is easy to work with. So we have some microwells, microfluidic system, and also the chips we have developed over the years. So I want to just talk about like a couple of different projects we just recently finished to help neurodegenerative community. And the whole point was based on the sequences which are associated with repeat disorders. Like all of the disorders based on the repeat sequences are neurodegenerative disorders like ALS or Huntington disease or myotonic dystrophy or MS. And this is because of the expansion of the different kind of repeat sequences like G4C2 for ALS and CGG for Fragile X. So our hypothesis was because of the expansion, there will be change in the 3D structure of the sequence or the properties, some biophysical properties, which we might be using for the detection scheme. So most of the work is actually is proof of concept, just to confirm that whether the expansion of the sequence itself will help us to distinguish between a normal gene versus abnormal gene. And the reason was because these sequences, when they expand, they make different structures. And based on the structure, their biophysical properties and interaction with other materials will turn into some different signal. So for example, this first one, I'm going to talk about the microfluidic corrector or the fluidic property of the CAG sequences associated with Huntington. The platform is pretty simple. We start with this PET substrate, design a channel, and print on this using Vex printer. And putting that into the sandwich of the microfluidic system and run the sequences. So what we found, we did like a lot of different characterization using different sequences, but I'm just giving you the results which are related to the detection of the CAG sequences. So what we see here, that increase in the length of this CAG sequence, their flow velocity is decreasing. So that might be because increase in length will turn into different 3D structure of the molecule or some physical property which impede its flow velocity. And this system does not require any external force. There is no external force applied for their movement. So we just drop the sample here and follow their movement, as we see on this chip. So even without applying any gel or any external force, we can see the difference in their fluidic character. And that's what we observe based on the different sequences as well as their length, as well as their concentration.
Can I have a question very quickly?
Yeah, go ahead.
This proof of principle experiment was just done with synthetic oligonucleotides encoding the region, or you done it with the whole gene, for example?
Okay, so this is synthetic, all right? This one is synthetic, and this one is the RNA containing CAG repeats, and it was from the neural stem cells, and they were generated from in iPSCs, right? So these are the sequences that we obtained from our collaborator, Professor Lisa Illerby from Bug Institute, and these are the RNA samples. So the previous one was, this is double-stranded DNA, and these are RNA samples. One has a length of 21, the other one has 72, right?
But
so
you don't pre-purify, if you wish, the RNA that encodes huntingtin. This is a whole population of RNA that you can run there and detect this, or is RNA isolated, specifically the huntingtin encoding RNA that you're putting in here?
Okay, so since I don't prepare sample myself, but I believe they have purified it.
Yeah, it must be. Yeah, because
so I think this is purified. This is a good question. So, but we also want to test some whole genome as well.
Yeah,
so down the road, that's what we want to test. That's our goal. That if the sequence is expanding in thousands and thousands of repeats, then that should affect the whole genome property. So we might be able to see the difference between the normal whole genome versus abnormal one, but we haven't tested that yet. So if we get one, then definitely we will test and then see whether we can see the difference. So now what we see here, RNA HD negative, which is only 21 repeats versus 72, because the pathogenic length, there is a debate on the pathogenic length for HD. Some say that is more than 30, some say it's more than 25, but the one we've got is 72, which is definitely a pathogenic length, and we see a clear difference between the normal and abnormal. If you just look at the error bar, although there's a significant difference, but again, based on the length difference, the difference is less. So we are trying to work on improving our system so we have better sensitivity and clear difference to avoid any false positive and false negative response. But the good thing is this kind of assay can be done in 80 seconds. And the volumes of the sample is just 25 microliter. Not 25, I think this is 0.25 micro. 25 microliter is too much. So it was even less than microliter volume. And that reduces the amount of sample, which is about 14 attomole. And one thing I just want to mention here, although we report like very exciting limit of detections down to attomole level, but when we try a real sample, we lose a little bit of sensitivity. But again, the sensitivity is good enough, which might be attractive for real samples. For example, if we have attomolar sensitivity in the real sample, we have sensitivity down to femtomolar, right? So it's still, it has a potential to apply a real sample. So the next project I'm going to talk about is about electrochemical systems, where we print electrodes on a PET substrate on this flexible device. Again, we just use two printers, Vex printer and inkjet printer. At the end, we drop molybdenum sulfide, which has very high affinity for DNA. And what DNA does, DNA actually amplify the current response. So DNA works as an electrocatalytic material, which amplify the current response of the surface. Since MOS2 has a high affinity for G, and then A, and then T, and then C. So our hypothesis was like, if the sequence is G-rich, it will have higher affinity for a molybdenum sulfide, and we will observe higher current response. I'm not going to show like all different characterization we did based on different sequences, but what we found that CGG has a very high affinity for MOS2. With respect to concentration, we measured from attomolar to micromolar concentration with the linear range, and the detection limit was found down to 3 attomolar. And with respect to the length, the sample was double-stranded. Again, synthetic here. This is synthetic. With respect to the length, using 10 attomolar concentration, we see increase in current with respect to length. However, when we have the CGG repeat DNA from plasmids, so we can see the difference between the shorter length and longer length. So for FXTAS, I believe the 26 repeats are the normal repeats, and more than 26, or I think more than 50 repeats for ataxia is abnormal sequence. So again, the difference is significant just by looking at the graph, but for the real analysis, we'll still need to improve the sensitivity, and we believe that if we further decrease the concentration, we might have no response from the normal and might have significant response from the abnormal sequence just based on the interaction between the CGG and MOS2 surface. So molybdenum sulfide is an inorganic material, which is 2D material, and it is just similar as graphene. Graphene, I think everybody knows about graphene, which is 2D carbon hexagonal ring structure. It also has very high affinity for guanine sequences, and almost similar trend as molybdenum sulfide. So the benefit of using graphene ink was we've pushed down the limit of detection down to zeptomolar level. So what it means is there's also potential for different materials to explore for sensitive signaling process and also some sort of selectivity. Now I will discuss the last project at this point, which we did for ALS community and detecting G4C2 sequences. And these expansions, they produce dipeptides. I'm going to talk about those dipeptides later because that's our future project. In this strategy, we just use two different type of probes. So the target might be really, really long. It might be like hundreds of repeats. It might be in hundreds. So, but we cannot have a probe on the surface which is like hundreds of repeats. So we thought, why not we just immobilize a small probe, like maybe one repeat or one C4G2, and here we have 3 repeat or 3 C4G2 repeats. So to look at the potential that if our target can bind to these probe sequences. So what we found that in terms of resistance, because when DNA is on the surface, it increases the charge, negative charge on the surface, which might result into the charge transfer resistance. The semicircle of this curve is a response which is charge transfer resistance, and we just translate that into number of repeats present in the sample. So if you just look at the response based on the P1, which has only one binding site, you can see this is a probe response only, and you can see a significant linear trend, like even we have 1 repeat, 3 repeats, and 6 repeats in the target sequence, which is again a smaller target. But again, we can see the difference based on the number of repeats present on the target, and this just small probe can capture that. However, the P2 repeat, which has 3 repeats, it is already saturated, and the response is not that distinguishable from repeat to repeat. Again, this is linear response, but this response is not that distinguishable compared to the one we have with single repeat in the probe. So we tested this probe on some real-to-life samples. For example, this is 12 repeat RNA, and this is non-specific CAG sequence, which was obtained from iPSC cells. And we see high response with the complementary G4C2, but almost a similar response as the probe for the non-complementary, which shows the specificity of the probe. So that was a success. And then we also detected the sequences like 30 G4 repeats and 450 G4C2 repeats from iPSC cells. So these were RNAs basically, and this is ALS negative sequence, this is ALS positive. Although we see a trend, but based on this response, the distinguished difference is a little bit compromised. So what it means is, like, we still need to work a little bit on the sensitivity of this platform and improve the capturing capacity of the system. But that proves the principle that we can detect these sequences by electrochemical impedance just by simple probe immobilization on a surface that will capture the target, even in the complex media and even in the presence of the non-specific molecule in the environment. So now we are moving on to start a new project on dipeptide biomarkers. So almost 7-8 years ago, it was reported like G4-C2 sequences, they produce 3 kinds of different dipeptide proteins, poly-GA, poly-GP, and poly-GR. The good thing is, like, both sense and antisense transcript, they produce poly-GP protein. So it increases the concentration of poly-GP in the cell, and it can be used as a pharmacodynamic marker. So you can see the difference in the amount of the poly-GP protein before or during or after the treatment. So the idea is simple. As I mentioned when I was discussing biosensing strategies, that you have antigen immobilized on the surface. So we have anti-C9OR72, poly-GP sense antisense antibody, which can be immobilized on the surface. It is available by Sigma and some other companies. And you bring your dipeptide target and see the response in terms of the resistance, or you can see the change in the frequency of the surface, or you can also see the change in the plasmon resonance. So we work on EIS, which actually measure the resistance of the surface. So so this is simple strategy we can develop to detect these dipeptide sequences. And so first aim is to develop the assay, which is first step to do, and then we need to test the sensitivity, selectivity, and also the cell extracted poly-GPs, as well as we are going to develop VEX on plastic chips for low cost detection, and that will be the last part of the project. And the benefit at the end is that it can be used in clinics, because doctors have to make decisions and they have to monitor in real time the amount of poly-GPs in patients' blood or its CSF. So that will be helpful, and they can make decisions based on the amount of poly-GPs, because the length of poly-GP don't vary from patient to patient. So each patient himself can be a control. So you can see the amount of poly-GPs before and after during the treatment. However, the length of the poly-GPs are not too varied from patient to patient. So that's what the idea is. So we are embarking on and trying to develop biosensing technology for poly-GP peptides. So here I will stop. Yeah, we did like almost 45 minutes, and I will be happy to answer any questions.
My plan for this is that we're going to eventually come up with a panel of biomarkers for ALS. And I think when we talked, you said we could put up to 10 different markers on one of these chips. So,
yes. So we are going to start with poly-GPs, but definitely every biomarker will need some preliminary work to develop sensing. And then we can have a bunch of different biomarkers in a single sample, and the surfaces on the chip will be modified with their selective, or you can say their capturing antibody or capturing molecule. So the chip is not a kind of big deal at this point. The first thing is to develop a protocol for the detection.
So a clinician, we might have, let's say we had be really lucky and get 10 or 15 biomarkers, then you might have 2 different chips because the platform on sensitivity might be different, or the analyte you're looking for might be different. You might be looking for a peptide repeat, or you might be looking for like neurofilament. That would require you to have 2 different chips, right?
Yes, yes. So, I mean, it can be one chip which can develop, which can detect like maybe 4, 5, or 6 panels, or you can have 2 which can have better capability. So, as I said, like developing chip itself or designing a chip itself is not a big deal. I would say the main thing is to develop a sensitive and selective protocol which we can translate or apply on a chip itself. So that is a thing, like using real samples and finding a selective and sensitive capturing antibodies or capturing molecules that can selectively detect those biomarkers.
And it would also be possible if we chose to downstream that if we knew that there was an efficacious treatment and we were concerned about the level, you would be able to develop a chip to monitor the amount of drug maybe in the serum over time, and maybe adjust or down as the patient needed, along with biomarkers.
So I think in that case, we need to integrate microfluidics with electrochemistry, that you run the drug. First you immobilize the biomarker on the surface, and then you run the drug and look at the response of the biomarker after interacting with the drug. So you can have some sort of like some preliminary test on a chip before you induce into the patient. So that can also be done. Second thing you can do is you get the sample out of the patient and then run on the chip, detecting the biomarker, the difference in the amount of the biomarker before and after the drug treatment. So that can also be done.
Okay. Anybody else got questions?
Yeah, very nice talk, Hamish. I was curious, like, from practically speaking, from what we would like to do, perhaps with you, is to look at blood samples. And I guess what we would envision is being able to have a blood drop, even with the red blood cells there, that could feed through your microfluidic system that would allow detection of, well, there's 2 ways I see this working. One is that you have an antigen, ideally a peptide, and we're trying to detect antibodies that are in the patient. This is not really so much for ALS, but for example, like detection of SARS-CoV-2 antibodies, that'd be really wonderful. But the other is that we're trying to do in this project is to identify a large number of antibodies that are actually markers of tissue damage in particular that could reveal an underlying pathology. So I guess your surfaces for, like how much antigen can you get on a surface if your capture agent is the antibody? Can we get the density that we need for using a single blood drop, for example, applied to your microfluid with an antibody?
I mean, that's a good question. I mean, the units for the proteins are used, they are different from the nucleic acid units we use. But in terms of the number of molecules, I would say just if I use molarity or moles, we can even go down to like few hundred molecules. And that also depends on the size of the surface. For example, using the surface of 2 millimeter versus 5 micrometer. So when you have a smaller surface, that increases your sensitivity. So you can detect even a smaller sample size on that because your surface is smaller. So we can go down to like few hundred molecules detection, but so before that, definitely we need to find very selective antigen on the surface to capture those antibodies. So I think as far as the density is concerned, density down to like a few hundred molecules or even less is not a problem. You can do that.
I mean, I'd be very interested in exploring with you to see if we gave you peptides, could we detect antibodies that are in a blood drop of a patient? That again has different applications, but for this particular project that we're doing of NDR, we're going to be looking for proteins that we don't expect to be in the serum in a healthy person. So we're looking for an increased level of detection of antibodies reactivity, well, of the antigens that we're going to be capturing with the antibodies on the chip. And so, I mean, if you're interested, we could work with you on that front, because we have a lot of antibodies and a lot of peptides. So we can look at both ways if you're interested.
Yes, we are definitely interested to actually screen many different kinds of antibodies and antigens to find the find the selectivity as well as their based on their affinity.
Right.
So there is a potential of screening the right antibodies and antigens for developing a very selective and sensitive protocol.
I mean, the real advantage of working with antibodies is that they usually don't detect other proteins that are in the serum, because
but there's always non-specific absorption because of the charge, because of the hydrophilicity kind of interaction. So that's what we always observe. And although theoretically sometimes we say, okay, they're very selective, but we lose sensitivity because of that non-specific absorption. And that's what we really have to focus on to mitigate or to reduce that non-specific absorption. So this takes a lot of time to develop a sensitive protocol.
Right. Yeah. Really nice talk, though. Really enjoyed it.
I've got a question. On all your slides, it seems you've just got one test you run. Is there potential in the future to have more than one test on a single slide so you can get combinations or analysis of ratios and things?
I think that's Siobhan was interested in, and that's what I think she was asking. People are doing it. So it's not something which is not achievable. The Abbott iStat I showed you earlier, their single cartridge can, one of the cartridge which can detect like 7 or 8 different kind of small molecules in the blood. So if you have a samples detection surface which has different kind of selective biorecognition molecules, and they bind to their own analyte, so it is possible that you can detect different analytes on the same chip. And that is happening. I mean, people are pushing the boundaries in that direction as well. And that's what we want as well, but it all depends like what kind of biomarkers we are interested in.
Yeah, I also have a quick question, like moving, I mean, first I thought it was a wonderful talk. I really enjoyed the basic principles and how you explain it to the lay audience. The one thing I wanted to mention, it relates to your detection of the dipeptide repeats, but then that would be actually very nice if we have to evaluate the efficacy of a therapy that is targeting the production of the peptides, for example. But I mean, and I don't know if somebody here in this panel will agree or with what I'm going to say. Maybe Steve actually agrees with this. It's like there's a lot of evidence now showing that in general terms for ALS patients, these neurofilaments proteins can be detected in cerebrospinal fluid. And I don't know about plasma. I think there's some reports on plasma, but I'm less sure about that. the idea is this is a very extreme, probably the most abundant protein in axons. And since the disease is associated with degeneration of axons, it appears that it seems to be a good marker. So that will expand a little bit the spectrum of ALS forms that you could evaluate and see how any given therapy is working. Have you heard about, I think, Steve, you're very familiar with very good anti-neurofilament antibodies, aren't you?
Yeah,
Not yet, but we are making them.
Yeah, well, there's a lot already available in the market as well, but it's good to know that you'll make some more. But I know those are very highly, they have very high affinity, many of them that I work with. They're really, really good antibodies. And it's something that it might be worth checking from the literature, because I would say that from I'm not as specialized on that, but from what I see, these neurofilaments do hold some promise, not just for ALS, but even for many other diseases that feature degeneration of axons. So it's just a comment about that you could probably have increased the number of variants that you could be looking how a given strategy is impacting that particular readout.
Yeah, I mean, I agree with you. When I and Shobhan were working on talking about a project, and NFL, it came up, and then she is really interested in, and we are also interested in. And that will be, I think, number of different biomarkers, including NFL and poly-GP proteins, which can combine together on a single chip and then detect a system for that. Yeah.
Okay. Well, thank you so much. That was great.
They do have, let me just say on neurofilaments that the MDS assay that Chandler's using and looking at, so you can measure them on a verified platform in mice and people. So Gerardo, even the mouse experiment that you're conducting now, if you've saved serum, you could run it on Chandler's MSD platform, save 0.25 microliters, it looks like, and give that to Dr. Shamsi down the road and see how they compared. So that's a doable thing. I think the only thing I think plasma is reasonable. I think from what I've read that humans will be negative early on, then go up, and then they hit a plateau where I think mice continue to go up over time, or it's the other way around. I can't remember, but I'm pretty sure it's humans plateau.
I will read more about that.
Hi, Matashin, I think a very interesting talk, and just a couple questions. Going back to the repeat detection. So when you look at like a CGG repeat or G4C2 repeat, are you considering the flanking sequence? So how do you really determine the specificity of those repeats that you are detecting? Because for example, like CGG repeat, there has been report now just came out like a couple weeks ago. There are probably like several thousands polymorphic CGG repeat in the human genome, but among the normal population. So how do you really distinguish, specifically look at like FMR1 CGG repeat?
Yeah, I mean, that's a really good question. So again, so this platform is not actually detecting the gene location. I think that's what you're talking about, because yeah. So these expansions can happen at different locations, and at different locations, based on location, they can have a different neurodegenerative disease, right? So if I just look at from that angle, we actually did not develop something to detect the gene location itself. The platform was just detecting the sequence and its length. So the main thrust was to detect whether the length is distinguishable or not. So we can have some sort of like yes or no answer based on that. Okay, so this is a normal versus abnormal. And again, the samples we got, they were highly purified, I would say, right? And if we just really want to look at the effect of the location, probably we need to work on the whole genome rather than the very selective and sequence, which is just very specifically cut off from the gene, and then we are detecting it. So
to a certain degree, if you can combine like a CRISPR-based capture
yes.
with your detections, then that would be, you
yes.
can really look at specific locus. Yeah, that's cool. And the second question is, in your detection, can you really distinguish methylated versus unmethylated, like repeat?
So based on literature, people say there's no difference between methylated and non-methylated sequences based on their electrochemical response. So what I work on their electrochemical property, the current they generate after interacting with 2D materials or the surfaces. So there wasn't any significant difference found between the current response of the methylated and non-methylated sequences. But I would say it also depends on the amount of methylation happening on the surface. The single methylation might not be, or maybe a couple of methylation might not have a significant effect. But if we have like large number of methylation, it might change the structure of the sequence, and changing the structure of the sequence might cause a difference in their interaction with other materials, which might transduce into a different current response. So we haven't actually tested that, but I believe the amount of methylation should have effect on the interaction of the DNA with the surfaces.
Yeah, I mean, I think in that case, I think the Fragile syndrome would be a perfect example. You can really look at Fragile syndrome, which is like with the CGG repeat hypermethylated. And then going back to the ALS, I think there's have been some reports indicating the methylation status of GPC2 could potentially modulate the phenotype as well. I think that's why I was asking whether you can really distinguish the methylation versus non-methylation, which might have the clinical utility as well.
Yeah, because people work on this methylation thing on a very synthetic and oligonucleotides, not like big sequences. That's why I didn't embark on, further exploring it. But based on the size of these expansions and their hypermethylation, I would expect definitely there will be effect on their current response for sure.
Anybody else? We'll close the meeting, and I'll go ahead and make a recording and then post where you guys can view it. So anybody else got any questions? Okay, well, thanks for participating, and thanks for giving us a great talk.
Thank you.
Talk. And
Thank you, everyone. Thank you very much.
and I think the next one, maybe we'll have some data to present. I'm not exactly sure, but I'll let you know before 3 weeks. Okay. Thank you.
Thank you.
All right.
Bye.
Bye.